Hello, my name is Jeff Barker. I'm a graduate student in the Hines group under Dr. Rochelle Hines. And today I'm going to be talking about my project, which looks at how regulated expression of the GABA A receptor alpha-2 subunit modulates the development of alcohol use disorder. Alcohol is a very prevalent drug in our society and the United States. In fact, I think a lot of people don't think of it as a drug because of the way it is heralded in society, and it's so ingrained into our culture and promoted through commercials and movies. But alcohol can become problematic for select people. You can develop an addiction to alcohol, known as alcohol use disorder. It can get in the way and interfere with relationships, hurt work productivity, cause a lot of health problems. And symptoms of alcohol use disorder would be also drinking, trying to stop and not being able to, starting through withdrawals upon quitting. And alcohol use disorder is very prevalent. There's around 14.5 million people as of 2019 in the United States alone that could be diagnosed with AUD. And at the present time, there's really not a lot of good pharmacological interventions for people with alcohol use disorder. My research aims to better understand the circuits in the brain that are involved with the development of AUD and possibly look for novel targets. That said, the general addiction pathway, there is a lot known about that. And it's the mesolimbic reward pathway, where upon consuming a drug, neurons in the ventral tegmental area are activated, and they project dopamine to these areas like the nucleus accumbens, which is my area of interest, as well as the amygdala, prefrontal cortex, and hippocampus. Now, alcohol is a sedative drug, and it acts on GABAergic transmission. So GABA is the major inhibitory neurotransmitter of the brain, and a GABA A receptor has 5 subunits, and they can be different combinations depending where it is and what its function may be. So some of them contain the alpha-2 subunit. And genome-wide association studies, where they looked at families of multiple alcoholics, they looked at their genes and they found a common single nucleotide polymorphism in the Gabra2 gene that codes for this alpha-2 subunit. This is kind of our first hint that the alpha-2 subunit may be important to the development of AUD. So in A and B here, we're looking at the nucleus accumbens. This is my primary region of interest, the main reward and motivation center of the brain that can eventually become hijacked throughout the process of addiction. The drug becomes the new reward. So we have on there neurons that contain D1 or D2 dopamine receptors. And the ones containing D1 are part of what's called the direct dopamine pathway. This is the one that's highly associated with the development of addiction. So we looked at neurons containing either D1 or D2 receptors and looked to see if they also contain or nearby alpha-2 receptors, alpha-2 subunits on a GABA A receptor. And you can see in A, there's a strong positive correlation with alpha-2 and D1 and a negative correlation with alpha-2 and D2, suggesting that this alpha-2 subunit is positively correlated with the direct dopamine pathway and possibly influential in addiction. And that's just at baseline levels. So we wanted to see if there are any changes on the development of addiction. We used, firstly, what's called the 2-bottle choice intermittent access protocol. So we use mice in our lab, and in this design, they're in a cage here, like you can see in A, where they have a choice between 2 bottles. They have either water or alcohol. And this is voluntary access, which is more valid and more like human consumption would be if we were to just inject that and see how things change. So we give them a period of 10 days of drinking, but we give them intermittent access. There's been a lot of research in the addiction literature showing that the particular reward schedule, how often you give rewards, can be a big influence on the development of addiction. In fact, our whole city, Las Vegas, is kind of built upon this idea, with slot machines where variations in reward cause people to keep coming back. This works for drinking as well in mice. So in C here, we're looking at the nucleus accumbens. And this is a heat map. And so the more bright reddish purple it gets, the more alpha-2 there is. And as you can see in the control, there's not a lot of alpha-2. But then after drinking and the nucleus accumbens, we see a large increase in alpha-2. And then we see that further represented in D, where both the core and the shell of the nucleus accumbens have higher alpha-2 levels after drinking. Well, this is another hint that alpha-2 is important in the development of AUD. Next, we wanted to see what would happen if we used our mutant mouse model, the Gabra2-1 mouse, that has saturated alpha-2 expression in the nucleus accumbens. So you can see these are some stains here looking at wild-type heterozygous and homozygous Gabra2-1 mice, staining for alpha-2. And you can see in the nucleus accumbens, this is at baseline levels, there's an increase in both the heterozygous and homozygous mutants. So behaviorally, we put them through the same design, the intermittent access two-bottle choice. And as you can see in A, they all start kind of around the same amount of drinking. And by day 10, the wild type is consuming a lot more, but the mutant mice are not. They never develop alcohol use disorder. They never develop a preference for alcohol. And you can see that reflected in B as well, where you just see day 1 and then day 10, there's a start where the wild type has almost doubled its drinking and the mutant has not really changed. Next, we did conditioned place preference, which is another validated protocol for addiction, where you have an apparatus that has several chambers. You give the mouse some time to get acquainted with the chambers, make some way to differentiate them, and you just kind of track how much time they spend in each room. And then eventually you will pair the drug with a certain room. So you give them the drug and put them in the same room each time, conditioning them to associate that room with the drug. And at the end of the test, you will not deliver the drug, and you just kind of put them in the apparatus and see where they spend their time. And an addicted mouse will spend their time in the conditioned room. They get conditioned pretty easily to recognize and associate that room with the drug. As you can see here, the wild type mouse spent a lot of time on the ethanol paired floor. So it definitely developed place preference, but the mutant mice did not. Once again, the het and homozygous did not develop conditioned place preference. So now we're looking into mechanisms that may regulate this change in alpha-2. A lot of therapies right now are receptor-based therapies, and the field is kind of moving to look at other possible means. And so I'm looking at this protein called the NONO protein. It's an mRNA binding protein that has the ability to affect transcription and translation, and ultimately can regulate the expression of the protein. And the NONO protein has been associated with alpha-2. So what we did here, these are Western blots. So these are mice that had been drinking, and these are different time periods. So yolk controls are mice that were put into the single housing apparatus but did not drink. And then there were mice that drank for a week, mice that drank for 2 weeks, and mice that drank for 4 weeks. And we see a very similar trajectory between NONO and alpha-2. Whereas as early as a week, we see they both kind of go up. And then NONO seems to go down first, and then alpha-2 comes down after. This trajectory suggests that there may be some correlation here, or it may be that NONO is regulating the change in alpha-2 that is regulating this development of alcohol use disorder. But just to kind of summarize, the GABA A receptor alpha-2 subunit is highly expressed in the nucleus accumbens along the direct dopamine pathway. It increases with voluntary access to alcohol. Our Gabra2-1 mouse model with saturated alpha-2 does not develop a preference for alcohol. And also, this mRNA binding NONO protein changes in tandem with alpha-2 levels. So we're working now using an adeno-associated virus to overexpress NONO and see how that affects drinking. We're just kind of in the preliminary stages now. I have a graph here, and it appears that the NONO mice also do not develop preference for alcohol, but we're still running more experiments, and we need to use some immunohistochemistry to look and verify expression and just check all that out. So thank you to Dr. Rochelle Hines, my mentor, Dr. Dustin Hines, and Reggie Anderson for collecting some of this data, and thank you for listening.